This is Bio 181, Chapter 15 video on transcription. So transcription is the first process described by the central dogma of molecular genetics, where we're going from DNA into messenger RNA. So we're going to take a sequence of DNA, we're going to break it apart. Remember, DNA is a double helix, so we're kind of going to break it apart. And we're going to look at one side of it. Over here would be like a T and an A and so forth. So we're going to look at one side as our template. And A, T, G. This is an example here. So this is my DNA. And then the RNA is going to be complementary paired by an enzyme called RNA polymerase. And we're going to make, if there's an adenine, we'll make a thymine. If there's a, oops, sorry. God, not, that's a good mistake. So in RNA, there's no thymine. So if you see adenine instead of thymine, it's going to pair with uracil. So that's a U. T will still pair with A. G will start pair with C. Adenine, again, with uracil. Cytosine with guanine. Cytosine with guanine. Thymine with adenine. So this is our messenger RNA. And you just keep going, creating a complementary strand of that information for your gene. And that's going to be used to make the protein. So the main steps are initiation at regions called promoter regions on the DNA, elongation by the enzyme and RNA polymerase. So again, a polymer is a long molecule made up of monomers. In this case, the polymer is RNA made up of lots of nucleotides. And then the termination sequence is going to be where the process stops. So I've got DNA. This is my color here. So here's my DNA. And I've got a sequence of the DNA that I'm reading. So the first region is called the promoter. And that's a sequence of DNA. The longer it is, the more likely that gene is going to be transcribed. There's transcription factors and things that can increase the likelihood of transcription. And then there is the gene itself. Okay. Well, the whole thing's the gene, I should say. This is going to be the transcribed unit, right? But this is the code basically for the protein, okay, that's going to be made, be modified a little bit. And then at the end, there is the termination sequence. So RNA polymerase is going to come along, right? Remember, that's our enzyme. And maybe I'll just make it green here, right? So RNA polymerase is going to come on. It's a big enzyme, polymerase. This is the same name we had for DNA enzyme. It was DNA polymerase. So in this case, it's making RNA. This red region down here, this is the sequence of DNA, the gene. And then, what else was I going to go over? Okay, so it's going to complementary pair. Right? So it will hop on here. And then again, it's going to start making the complementary pair. So maybe U, C, C, U, A, C, C, G, whatever that is. So a long sequence. And then it will hit the termination sequence. And then our RNA polymerase will leave. And you've got your initial mRNA transcript. This is called a pre-mRNA transcript because it can be modified. But this is what we're starting with. All right. So the transcription unit is the sequence between the initiation and termination sites. And so you can see here, the whole thing is the gene. You've got a promoter region. This is my DNA down here, right? You've got the coding sequence for the protein, and then you've got your terminator region. Oops. All right. Okay, so we're modifying the primary messenger RNA transcript, or that's often called pre-mRNA. So that's going to be modified. So here's my pre-mRNA. Again, I've got my A's and U's, C's and G's. So we're going to modify it and stabilize it by putting on at the 5' end and at the 3' end a cap on the 5' end. Got a 5' cap. And then we've got a poly A tail. And just like it sounds, poly being many, you're just adding on a whole bunch of these residue bases at the end. Okay, so this is called the poly A tail. Right. Okay, so we do that, and then we're going to remove sections called introns. So the whole sequence is not going to be utilized. So there's going to be some sections in it that we're going to cut out. So we cut out introns. And the enzyme complex that's going to do that, let's see, we've got this complex here that's going to cut this out and rebond this section. That's called the spliceosome. Okay, spliceosome. All right, so that's the overview. RNA splicing, pre-mRNA splicing, it's a post-transcriptional process. So that just means it happens after transcription, right? So after the DNA is copied to the messenger RNA, remember that's called transcription. So it's after that, and we are still in the nucleus. So we're in the nucleus in eukaryotes and prokaryotes. Again, this is going to happen in the cytoplasm. And we're going to take out introns from the transcript. So we're going to remove parts. So you've got this long transcript, and there's several sections that can be removed. Those are called introns. The spliceosome will then reanneal it. So that is pre-mRNA splicing. If you've ever taken two wires and cut out a piece that had a short in it and put it back together, that's splicing together wires. It's a similar thing. The spliceosome, that's that RNA protein complex. So it's made out of a lot of different molecules. We're not going to worry about it too much, exactly what's in there. There's these SNRPs that are part of it, but it's going to assemble, cut out the introns. So this is the spliceosome, right? This large complex made up of RNA and proteins that come together. Okay. And it's going to cut out the introns. So they can be cut out in different ways, and this is going to create different proteins. So you've got from a single initial transcript, you can actually make several related proteins like smooth muscle or skeletal muscle by what you remove and what you leave in. So exons, those are the things that are going to exit the nucleus. That's how I think of it. Those are the things those are the sections you leave in. Okay. So you leave in the exons, you take out the introns. So if I was over here and I had, well, actually, here we go. So introns are going to vary based on what you're taking out for specific protein. So here's my original sequence of DNA. These are all possible regions that could stay in while the introns are taken out. And so you could see that in the first example here, we're only keeping 1, 3, and 5. So we're keeping those exons. And we've gotten rid of the other introns. And in this case, we didn't keep in 2 or 4. So this is showing that this region of RNA kind of created that section of the protein having a characteristic shape. This region of RNA created this section, and then here we have this region of the protein. So that's one protein that could come from an original sequence of DNA. Here's another example, protein 2, right? It still has the same shape for section 3, but it now also has a few additional complements, 4 and 2. And then over here, similarly, we've got 2 and 5 and 1 put together. So you can have more proteins made than genes. So it was kind of surprising initially to scientists to figure out we had less genes than was predicted because you could use one gene to make many proteins. All right, and that's it for this video.